coastal environmental science and is currently pursuing a master's degree in science and soil and water science at the University of Florida. Her research focuses on water quality and the associated biogeochemical processes found in the local intracoastal waterway. In the Osborne Lab at the University of Florida, she contributes to iCoast, an initiative that seeks to create a database of sensing information that will allow real-time management of threats to the natural and built environments of the coast. As an iCoast team member, she calibrates and deploys 12 YSI XO2 water quality sondes and manages the collected data. And today, Taryn will be sharing with us the water quality dynamics in a tidally influenced backwater creek along rural urban gradient in Northeast Florida. And Taryn, you should have control over the slides now. And it looks like you're still muted. Or maybe your microphone isn't connected if you changed microphones.
Hello?
There we go. Great. Thank you, Taryn.
Okay, that was weird. Okay, thank you for the introduction. So hey everybody, my name's Taryn. I'm a master's student at the Whitney Lab for Marine Bioscience here in St. Augustine, Florida. And today I'll be unfolding the story of my thesis research looking at the water quality dynamics in tidally influenced rivers along a rural urban gradient here in Northeast Florida. So this begins with me really wanting to understand how urbanization is affecting our local water quality. So here I have a diagram providing some background information on how urbanization can affect water quality. So in a natural or rural system, rain is going to infiltrate the soil, and we don't see a whole lot of surface runoff because of that. And this infiltration combined with high percentages of forest cover leads to high dissolved organic matter inputs, or DOM, which is crucial for biogeochemical cycling. And then in a suburban setting, we see more impervious surface coverage. So that's roads, houses, sidewalks, and so on. And these impervious surfaces are going to decrease the infiltration capacity of the soil and therefore increase the volume and speed at which overland surface flow enters our local waterways. And that's going to bring in also sediments and pollutants as well, potentially. And so this is an example of what I have called here as hydrological flashiness. So then with this, we might also see greater inputs of nitrogen and phosphorus due to increased fertilizer use and other anthropogenic inputs. And from there, we might see lower dissolved oxygen because of the process of eutrophication. And we might also see greater fecal indicators. So then in the urban landscape, we might see, we'll probably see more impervious coverage and also more connectivity of that coverage. So then that might, again, influence the hydrological flashiness and make that even more pronounced. And then we also might see greater air and water temperatures due to that urban heat island effect. So the University of Florida's Provost Office funded a water quality monitoring initiative called iCoast in 2019. And this allowed me to establish several different monitoring sites here in St. Augustine and the GTM region. So this map on the right shows all of the sites that I used for my thesis research. So I focused on three main rivers. And so I focused on San Sebastian, which is next to downtown St. Augustine. South of San Sebastian is Mulcher Creek, and this is bordered by two residential neighborhoods on both sides. And lastly, Palliser Creek, which is the rural or pristine creek that I used as sort of a control. And this is bordered by a state park and a preserve. So the sites that I used are a combination of the GTM sites and the ICO sites. And so also the sites that are marked with a little star inside the red dot are those initial sites that I'll be referencing throughout my presentation. And then after my initial analysis, I started to consider the rest of these sites, such as a site at the Matanzas Inlet, a site at the Whitney Dock, and also the GTM sondes that are also in these study creeks. So at all of these sites, multi-parameter water quality sondes are deployed. So these sondes that are shown on the right take measurements every 15 minutes continuously, and I deploy a newly calibrated sonde every 2 to 3 weeks to switch them out and make sure that there's limited sensor drift. And the GTM and iCoast initiative use the same YSI XO2 sondes, and they also take measurements both at 15 minutes, so it's easy to incorporate all of these sites together. And so these YSI XO2 sondes measure temperature, turbidity, dissolved oxygen, salinity, chlorophyll A, and pH. And one thing that sets apart my XO2 sondes from the GTMs is that I also measure fluorescent dissolved organic matter, or FDOM. So I took measurements from May to December of 2020. So for my 3 initial sites that I was looking at, or the 3 creeks, I classified them as urban, suburban, or rural based on land cover percentages that were compiled by Trisha Kaisar, who has already been mentioned quite a few times today, and she's a PhD student at UF and collaborates a lot with the GTM. So this first row is showing the percentage of urban land within each of these watersheds. So we see that San Sebastian has the highest percentage of urban land at 57.1%, followed by Moultrie Creek. And lastly, Palliser Creek has the lowest percentage of urban land at 4.8%. And then the second box is the percentage of upland forests. And we see that San Sebastian has the lowest percentage, and Palliser Creek has the highest percentage at 66.5%. So my initial analysis of these 3 sites was very overwhelming, to say the least. And this is an example of one of the very first graphs that I've made with the data that I collected. So I really want to point out the variability here between sites, and this is mostly due to all the confounding variables that are in a monitoring study in a coastal area. So salinity, tidal influence, river morphology, and so on. So these differences that we see here aren't necessarily attributed to urbanization. You know, there's so much going on, so much noise. So it's impossible to attribute what we see here to urbanization. So this kind of visualization, though, can help identify some significant responses that a particular site may have. So I use these crazy graphs to pinpoint some certain events where I could then take a closer look and get some further direction. So these graphs on the next 2 slides are very crazy, so I'm only going to focus on a few key points here and then move on. So don't spend too much time getting yourself in a tizzy because of everything that's on these. So the next part of my story involved picking out a couple of weather events, such as the nor'easter event that happened last September, September 19th through the 21st, and also a few rainfall events in the beginning of November. So the graphs on this slide are showing each parameter's response for each site during a 10-day period surrounding the nor'easter event. And we see some examples of sites having unique responses following these events. So in the upper left-hand corner, we see turbidity, and I have circled where we can see some significant responses. And this orange line represents Moultrie Creek, that suburban site, and the purple is the Matanzas site. And then when we look at the bottom right, this is a graph of FDOM over that 10-day period. And again, we see a response from Moultrie Creek and also a response from the Whitney Dock site. Okay. So I also focused on the 3 consecutive rainfall events that occurred in the beginning of November. And we don't see as drastic of a response here from the sites, but as many of you might remember, if you were here during the nor'easter, it brought sustained high tides, rainfall, and wind for that period of 3 days. So when compared to these rainfall events only brought about 2 centimeters of rain over the course of 2 to 3 hours. And of course, they're separated by a couple days in between. So we don't really expect such a dramatic response from the sites based on these rainfall events. But focusing on these specific events allowed me to see how each site might respond differently. But there's obviously still so much going on within these data sets. So next I made scatter plots of the sites for both storm events. And I used specifically the sites that had FDOM sensors. So Matanzas, Whitney, Moultrie, Palliser Creek, and San Sebastian. I do want to point out that San Sebastian is not included in the nor'easter analysis because it had not been deployed yet. It was deployed in late October, so it is included in the November analysis. So in this slide and the next one, salinity will be plotted against fluorescent dissolved organic matter on the left side, and salinity will be plotted against turbidity on the right side. And just by looking at this, we can see that Moultrie Creek is much more has much more variance compared to the other sites, and same with the turbidity. And then looking at this table at the bottom, this shows the standard deviations and means for each of these sites. And we see that Whitney and Moultrie Creek have the highest standard deviations, and Moultrie Creek also has the highest standard deviation of turbidity and the highest mean. So then the similar results came from the November events, where we see Moultrie Creek and Whitney having the highest standard deviations of FDOM and turbidity. And we also see that Moultrie Creek has the highest mean turbidity. So this is not necessarily tied to salinity. So this higher mean and variation could potentially be due to that hydrological flashiness that I was talking about before, but we don't know for sure. So next we decided to run a principal component analysis to determine which of our many variables are causing the most variation between our sites. So this is the crucial piece of our story. These PCA analyses included those initial 7 parameters that I was looking at, but they also included the month, the average monthly rainfall, the distance to the inlet, and the distance to the freshwater source. So this PCA depicts those initial 3 sites that I was talking about, and includes all the data points that I collected from May to December. And we can clearly see that San Sebastian and Moultrie have some overlap, and they're pretty different from Palliser Creek. And we see that Moultrie Creek also has this spread in the upper right-hand corner. And then we introduce the biplot showing the polar contribution of each of the variables. And we see that the distance to the freshwater source, the distance to the inlet, salinity, pH, and dissolved oxygen are what's causing this huge divide between San Sebastian and Moultrie from Palliser. And we also see that turbidity is contributing to the spread up here. So then next, I included the rest of the sites, and I used average monthly data for all the parameters instead of every collected data point.
3 minutes.
And so the biggest takeaway here is that we see the saline sites are all clumped together and pretty distinct from the freshwater sites. And then Moultrie Creek is right here in the middle. And looking at the biplot with the variables, it basically shows what we saw in the first one, where the distance to the freshwater and saltwater source is contributing to the most variation, as well as like salinity, DO, and pH. And all of these parameters are very tightly linked. The tidal influence is obviously affecting salinity, but it's also introducing dissolved oxygen and buffering the pH. So I also measured total coliform, which is in the dark gray in this bar graph, and E. coli, which is the striped white and gray. And I measured these monthly for those 3 initial sites with the prediction that these fecal indicators would increase with urbanization. So we expected Palliser to have the lowest amount of fecal indicators and San Sebastian to have the highest. But we actually see the opposite, but this is most likely due to the location of, or the placement of the sondes. So we already knew that Palliser Creek was impaired for E. coli and fecal indicators. So it's no surprise that we see the opposite there. But with San Sebastian, we might not be seeing higher values here because it's closer to the inlet compared to Moultrie. So it's seeing greater tidal influence, and maybe that's reducing the residence time of these fecal indicators. So with the graphs that I just dumped on you, the story points to a major idea, or what I like to think of as a major lesson, and that is that the site locations relative to the distances to the fresh and saltwater is critical for being able to detect any effects of urbanization. So where I have deployed the sondes in each of those rivers are vastly different from one another in terms of their proximity to the freshwater or the inlet. And I have that expressed in this plot here, where all of the sites relative to their distances to the fresh and saltwater source are plotted. So for instance, my Palliser Creek sonde is very close to the headwaters and very far away from the inlet. So we're seeing much more FDOM compared to the other sites. And then the Matanzas and San Sebastian sondes are very close to the inlet compared to the others. And therefore we can't see a lot of discrete effects if the tides are carrying them away. And we see the most parameter response variation in Moultrie Creek, which, as you see, is the site that's positioned in the middle of all of them. So the takeaway from this is that optimizing your site locations in coastal studies to minimize the buffering that occurs at either end of the spectrum is important because we want to be able to minimize that buffering that's there to determine the effects of urbanization. So although we didn't determine the effects of urbanization in this region, understanding where to put our sondes is crucial for being able to move forward in order to do so, especially with the parameters that we were focusing on. So measuring flow data, nutrients, and so on would have made this a little bit easier. But obviously with resource constraints and COVID and everything, it wasn't exactly possible. So I'd like to thank UF and the Soil and Water Sciences Department, and of course my lab for all their support and guidance, including my advisor, Dr. Todd Osborne, our lab manager, Anna Thornton, and our postdoc and past student, Dr. Tracy Schaefer, for her guidance as well. And I'd also like to thank Trisha Kaisar for providing the GIS data. So yeah, I'd happily answer any questions. Thank you.
Thank you so much, Taryn. We do have several questions in the chat box for you. The first is, why do you think that the Palliser Creek site, or the Palliser site, is so smooth compared to other sites, which seem to have a regular cycle, presumably tidal?
So the Palliser Creek sonde that I have out there is very close to the headwaters. So there isn't as much of a tidal influence there compared to almost every single other site that I have out there. So when you look at the salinity of Palliser Creek, it's almost always very close to zero. So that's why most of the parameters are basically staying the same throughout the time period.
Great, thank you. This is 2 questions. First, where is the sonde in the San Sebastian, and do you plan to keep it in the water regularly?
So the San Sebastian sonde is at a marina. I don't remember the name of the marina, but I was there and they said that past Flagler students had placed a sonde there. So I'm assuming you know where it is. But in the future, I am planning to keep it there, I think. I mean, ideally, I want to have a sonde at a downstream site and one at an upstream site, so you can make a comparison there. So I'm hoping it'll still be there. And then
Was it potentially the River's Edge Marina?
I have no clue. I should
That's
probably know.
okay.
I have the business card. I just didn't think to look at the name.
Oh, thank you so much. Are you collecting regular samples for nutrient and coliform bacteria analysis? And do you think that the San Sebastian River gets any fresh water? Or, yeah, do you think it gets any fresh water other than rainwater?
Hmm. I do think it gets mostly rainwater since it's so far downstream. Um, I think a lot of the fresh water that enters from the headwaters is already mixed with the tidal influence pretty far up from where I was measuring. And collecting regular samples for nutrients. So I do want to do this in the future. I didn't take any nutrient sampling samples, but I do want to continue taking the fecal indicator samples, maybe doing that before and after rain events to see what happens there and if rain events are actually bringing in more E. coli after due to this like hydrological flashiness. But I also want to start collecting nutrient data as well. I've mostly just been relying on the GTM's monthly nutrient collection. So yeah.
Great, thank you. And we have time for one more question, but there are some additional questions in the chat box, Taryn, that maybe you'll be able to answer after this question. Do you plan to move the sondes given what you found, or maybe ask a different question with the data?
Yes, I mean, if I'm going to continue this, I would definitely move them to answer the question that I'm hoping to answer, which is, is this hydrological flashiness affecting the water quality or other aspects of urbanization? So, like I said before, I think it would be ideal to move the sondes further upstream and have them further away from the inlet because I think the inlet has more of an influence compared to even the freshwater inputs far up. So I think it's important to have the sondes within each river at comparable distances from both the freshwater source and the inlet. So I would then have to figure out what the optimal location for those are. I haven't really looked into that much yet, but there's also the problem of like accessibility, which is the reason why I chose the sites that I did, because I had to be able to walk out there and switch them out. So that's an additional problem with finding the best site.
Well, Taryn, it sure sounds like you're going to stay busy with this project in the future. Thank you so much for sharing it with us.
Yeah.
And if you do have time to stick around, there are some additional questions in the chat box for you.
Thank you so much.